Hi, this is Miss Linton, and this is 7th period review. Say hi.
Hi.
And we have 2 chapters we're reviewing for. I don't know why I did that, but I did that. So we're going to start with, I'm going to make sure I'm done at 3. Okay. So we're going to start with chapter 27, which is the origin and evolution of life. But we kind of start with the whole planet system as well. We review that as well and a little bit of plate tectonics. So the key things I would want to know about this is we're trying to get into the idea that the different parts, we have the core, right, hot. We have convection currents that move what? The plates around, right? Remember continental drift? Remember how we talked about that? Got evolved into plate tectonic theory. And the plates move. What happens if 2 plates go towards each other? What could happen? What happens if 2 plates go towards each other? Yeah, you could make a mountain, or one could go under the other one, like an oceanic plate could go underneath a continental plate, and then you could get a volcano, right? Or they could go apart from each other as well. Do you remember that also from FOSS last year? Yes, because we studied all of that. You remember. We talked about how, we talked about absolute dating. We talked about how isotopes break down to a more stable form, and that releases energy as well. We talked about those convection currents that are moving the plates above it. We looked at 3 different plate boundaries. Now, what happens at a convergent boundary is going to largely determine on what kind of plates are converging. If it's 2 continental plates converging, you're probably going to build a mountain. You could, even with 2 oceanic plates, build a mountain slowly, slowly, slowly until it comes up out of the water, right, and then forms an island, yes? But if you have an oceanic plate and a continental plate meet, what's going to happen? The oceanic plate's going to go under the other one. Now, here at divergent boundaries, where do we see divergent boundaries? At like oceanic ridges, do you remember that? Okay. Did we see it anywhere on land? Yeah. Okay. And then transform boundaries, that's what we're concerned about here in California, right? Because we've got the Pacific plate and the North American plate going past one another, and those are causing fault lines, little weaknesses, right, as it spreads out, and those cause what for us? Earthquakes. Okay? So you just want to review your 3 plate boundaries, and then know what's moving those plates are the convection currents, right? In the mantle is what's moving those plates. All right? And we looked at evidence for the plate tectonic theory. We looked at how we could look at the volcanoes. We could look at the way that at these upwelling in the mid-oceanic ridges that you can see that this is the oceanic plates are newer than the continental plates. We looked at the magnetism. We looked at the weight, like you can see that the continents are like floating on the magma below it. We looked at depression caused by large bodies of water. We look at how all the continents fit together. And there was one supercontinent, Pangea. Was that the first supercontinent? No. Will that be the last supercontinent? No. Okay? It's just going to take a little bit of time, and we can see how they all fit together. And you could look at species of fossils that are continuous amongst those continents when they were together. You can also see things like reptiles, which need warm, right, air, being in Antarctica, right, in those fossils. And the only way it explains that is if either somebody flew a whole bunch of reptiles and dropped them, or Antarctica was in a different location, right, and those organisms had adapted to that place. All right, so then we looked at how the Earth has changed over time, and one of the big things we talked about was the generation of what gas? You're breathing it right now. Oxygen, right? And we talked about cyanobacteria probably being the first ones to generate that gas. And by having that gas present, you could go from being unicellular to multicellular, right? And that you could go to living on land. Okay. So then we started talking about the whole story of the Earth and how it started. And we talked through how at first there was just the gases, right? And then those condensed due to gravity about how long ago to form our Earth? How many years ago? 4.6, how many? Billion. 4.6 billion years ago, right? Compression, you started having what? Volcanoes that were releasing gases out there. What was missing from the environment that is here now? Oxygen. So it's called a what kind of environment? Reducing environment. And it's very conducive to building. Now we don't have a reducing environment, we have a oxidizing environment that is not conducive to building. And that's why you don't see new things like evolving up out of the waters right now, because it's a totally different environment than when life first started developing. And we looked at the steps of what would have to happen. We said that inorganic molecules like these that you could find coming out of volcanoes or at hydrothermal vents, they would have to combine to form organic molecules like these. And we said, I wonder if that could even happen. A man named Operin hypothesized it, but who is the one that actually did the experiment? What was his name? Miller's. Like, you know, okay. And so Miller did an experiment in just a little over a week's time. When he circled, here's the experiment, when he circled these early gases in just a little over a week's time, with this right here, heat representing the, what did it represent? Molten magma, and these electrodes were representing lightning. And you know you had radiation from the Earth's core and UV radiation from the sun. You had all those sources of energy, and with these gases here that were on early Earth that came out of volcanoes or from hydrothermal vents, you were able to form these simple organic molecules. And then from these simple organic molecules, then you would build more complex organic molecules. And there are molecules like those present. Those are protenoid microspheres. When hot proteins cool, they start to form a sphere. Or fats make these fat droplets. So when we looked, and this is the picture that's in your book, I believe, is you have early Earth, which is about 4.6 billion years ago. Okay, and the first life forms are about 3.5 billion years ago. So a billion years worth of time had to take place on our Earth before this happened. Well, going from these inorganic molecules to small organic molecules, Miller was able to do that in just a little over 1 week, 1 week's time. Then you would have to put these organic molecules to build the macromolecules. And again, you have that same energy. We also talked about puddles and on clay being enzymatic to catalyze those reactions. And then you have your first cell. You need some sort of barrier, a cell. What goes around the outside? A cell membrane. So what's going on inside, the interior of the cell, is different than what's going on outside. And then once it's self-replicating, has a way to store things like DNA and RNA, then you would have a true cell. So when we look back through here, inorganic to organic, what energy did we have to make that step to build those molecules? We had molten magma. We had what? Radiation. We had lightning and UV radiation from the sun to help us build these. Then to build these macromolecules, these polymers like complex proteins and carbohydrates and lipids, then we had the same energy, but they could have been in little pools or on the surfaces of clay, which can act enzymatic. And then you eventually have your first cell. Was your first cell probably prokaryotic or eukaryotic? Prokaryotic, okay, because that's the simpler cell, right? And then was your first cell an autotroph or a heterotroph? A heterotroph. It's easier to consume nutrients than to be able to take energy and build sugars and then break them back down. So the first ones were prokaryotes and they were heterotrophs. I can't fit that on there. Then when the oxygen revolution occurred about 2.3 billion years ago, so another billion years passed, all of a sudden there was oxygen around, and you could go from being unicellular to being multicellular. Okay. And then that was it for chapter 27 that we needed to know. Okay, and that's the one that's probably the farthest away from your memory. Are there any questions, or ask your bio buddy? Is there any stuff that you need to go back over on that? You want me to ask them to go to the next one? I'm looking. Oh, is this it? Yep, it is. Okay, so then, any questions you want to ask me before I leave that? Yes?
So prokaryotics, they came before, so then eukaryotics
Came later.
Later.
Right. A eukaryotic cell is larger and more complex. And I'm going to teach you later. I didn't teach it to you now. I don't think the endosymbiotic hypothesis. Now one prokaryote lives in. Okay, so don't need more about that. Eukaryotic cells came later.
Okay, so that's what we need to know?
Yeah.
So pro first, you later.
What else? More questions? Okay, so let's have some closure, okay, on chapter 27. Structure of the planet, core, mantle, plates. Plates move. It's called the plate tectonic theory. Why did they move? Because of the convection currents in the mantle. They move, that affects what life can be on them. Oxygen evolved, that helps what life can be on them. Second thing, we talked about the 3 different boundaries, right? Then we talked about the evolution of life on Earth. And we said we had gases coming out of volcanoes. We had energy from the sun, from radiation, from lightning, from UV radiation. And using that energy, we could take inorganic molecules and make organic molecules. Using that energy and clay surfaces, which acts like enzymes, and puddles, things evaporating out and getting warm and hot, we could maybe build some complex molecules. And from those, we could build our first cells. Our first cells were prokaryotic, and they were heterotroph. Later, we had autotrophs evolve and photosynthesis evolve. Now we have oxygen. Now we can go from being unicellular to being multicellular. That's it in summary right there. Okay, being comfortable with what I just said. Now we're saying, hey, the study of life, so what do you consider life? Well, what are the characteristics of life? Let's do those together right now. Here we go. Living things are organized, made of cells. Let's talk about how they're organized. It goes atoms, molecules, organelles. Do all cells have organelles?
No.
What cells have organelles? Eukaryotic. You are eukaryotic. Otherwise, it would just go atoms, molecules, cells. And it would stop for prokaryotic cells because that's all they are. They stop at cells. They don't go any higher than that. Okay? Again, atoms, molecules, organelles, cells, because this is where life starts, tissues, organs, organ systems, organisms, population, community, ecosystem, biosphere, where all life exists. Living things are organized, made of cells. They acquire materials and energy. They maintain homeostasis. They respond to stimuli and reproduce. They grow and develop, and they adapt and die. Perfect. Okay? And then we said, how are they organized? Atom all the way up to biosphere. Do you think it's important for you to know that order? Yeah. Do you think it's important for you to know the characteristics of life? I agree. Okay, so I gave you a bunch of different examples of that. Responding to stimuli could be behavior. Responding to stimuli could be following the light like a plant does, right? And then we talked about emergent properties, which would mean, right, a multicellular organism can do something, can do more than a unicellular. You have different properties. An organ system can do more than an organ. Communities, you have more complexity in communities than you do a single population. Those are called emergent properties as you go up. One of the things we talked about was adaptations. And change over time is evolution. The mechanism for that, most of it, would be probably attributed to natural selection. Why are you changing? Well, because you're not going to survive if you don't. So there will be mutations that occur, and those mutations that are beneficial are going to be kept, right? Why did giraffes' necks get longer? Because they stretched them their whole life? No. Whichever giraffes had the longest necks got to eat the most food, and they were the biggest and the strongest, and they probably did the most reproduction. And of their offspring, They had ones with a little bit longer necks and some who had shorter necks. But of their offspring, which ones were going to live? The ones with the longest necks, right? And then of their next generation and their next generation. So evolution is change over time. The mechanism for that is natural selection. Who's selecting who lives and dies? Nature. Okay? There are other things that can affect it as well, okay? But that's the largest mechanism for natural selection, is those adaptations. So then we said, we know what life is, let's classify it. So we had classification categories. Do you remember the little song I told you, remember? Domain, then what? Kingdom, phylum, class, order, family, genus, species. And we said, did King Philip come over for good soup? Then we said, let's learn just one classification, top to bottom. What is the most exclusive word in classification? What is the most exclusive? Exclusive, you're excluding. Species. What is the most inclusive? Domain. Most inclusive is domain. How many domains are there? 3 domains. What domain are you in? Eukarya. Domain, eukarya. Kingdom, Animalia, because animal kingdom. Domain, kingdom, phylum. Phylum, we remember PC, chordata. Domain, kingdom, phylum, class. What class are we in? Look at all the mammals. Class, then what comes next? Order, how do we remember that? No monkey. So it's order, primates. What comes after order? Family. The family is Hominidae, okay? And then genus, species is Homo sapien. So I would know that because I might ask you one of those too. I bet I would, okay? And we looked at how it's becoming more and more and more exclusive, right, as we work our way down. We reviewed the 3 domains. These two have something in common. What do they have in common? What do they have in common? Look at the words that describe them. They're all prokaryotes. They're all the simple cells. Okay? Bacteria and archaea are both prokaryotic. No membrane enclosed organelles. You are what? Eukaryotic. The eukaryotes have 4 kingdoms, right? This is a domain. It goes domain, then what? Kingdom. What kingdom are you in? Animal. What's another kingdom? Plants. Good. What's another one? Fungi. What's another one? Protista. Okay? Nutritionally, animals are what? Heterotrophs. You know this, right? We learned this. We did a whole unit on it. Wow. What kind of heterotrophs do we call them? Ingestive heterotrophs. We have to bring food in. What's another kind of heterotroph we learned about? What's another? There's an N in fungi. What's another one? Absorptive heterotrophs. What would plants be called? Autotrophs. Protista are both of those. Usually unicellular, but there are some multicellular ones. Okay, so these are all in the domain eukarya. What sets apart bacteria and archaea? Archaea is actually more closely related to eukarya. Bacteria is the oldest. But what's another name for these guys? They're what? Extremophiles. They live in really extreme conditions, like where it's really salty or really hot in temperature. Okay, and we looked at the different kingdoms. We said binomial nomenclature, you use the genus species name in italics. We talked about, this part was reviewed. The biosphere is where all life exists, water, air, land. We reviewed the words ecology, population, community. We reviewed these words. That should be familiar to you. We talked about how energy flows and nutrients cycle. We reviewed that man oftentimes is detrimental, right? Modifies ecosystems for its own purposes. We know all the cycles and we know how man screws those up, right? Okay, but we don't always have to. We talked about the importance of maintaining biodiversity, a bunch of different life, both genetically, having genetic biodiversity amongst us, right? Having various species and various places to live. And we will end our year going back to the importance of that. Then the last thing we talked about was the scientific method, okay? And we talked about the processes of how you do labs, right? And we talked about qualitative and quantitative data. Qualitative data is what? Give me an example of qualitative data. Color, okay? Whereas quantitative data is always going to be a what? Number. Good. Okay? Independent, dependent variables. Independently you choose those. Dependent is data. We talked about our science practices. You don't have to sit there and memorize the science practices, right? I'm not going to like list the science practices. I'm just showing you, when you look at your lab, do we ask questions? Do we use models? Do we investigate? Do we interpret? Do we use mathematics? Remember how we looked at our just last lab and we do all of those things. And then the last thing we just talked about was science and social responsibility. We talked about bioethics. And the question we wanted to know is, we have all these different technologies, but just because we can do those things, should we do those things? How do we decide? Society has to decide. Science doesn't decide, and scientists don't decide. They have the technology, but we decide how we use it. We use it for good or evil. That's up to society to decide. Okay, questions or concerns? Yes?
Can you go over the scientific method again?
Sure. So on your scientific method, we talked about the first thing you do is you make observations, right? Like what's going on here. Okay, and we start to ask some sort of question based on our observation. We do some research. We want to have a little bit of, we have this phenomenon. We ask a question about it. We do some research to see how much we do know about it currently, and then we can put forth a hypothesis. Once we have our educated guess, our hypothesis, that's, remember we do if-then. Remember we talked about the different kinds of null hypothesis, a research hypothesis. Then we can do an experiment. The two words, controls and constants, or control or constants. Constant is everything we keep the same, right? The control is our basis of comparison. If I want to know how fertilizer affects plant growth, my control would be plants without any fertilizer. Okay? That would be my control. My constants is I give them the same amount of water, I plant them the same depth in the soil, they get the same amount of sunshine. Those are constants for every single thing that I'm doing in that experiment. My control would be the one without any fertilizer, so I can see how fertilizer impacts plant growth. I analyze my data, oftentimes using math, and then I come up with my conclusions. And we talked about claim, evidence, reason. Okay, anything else on that? Okay, review. Do your additional objectives. You have 2 chapters worth of additional objectives, and I hope you have some toast.